I'm excited to discuss with you today the role of the innate immune system in cerebrovascular injury, and more specifically, how different innate immune cells at different times may have positive or detrimental role in this process. There are multiple neurosurgical disease entities that have cerebrovascular injury in common. These include and are not limited to traumatic brain injury, primary intracerebral hemorrhage, and ischemic stroke. All these entities have in common that following the initial insult, there is associated secondary parenchymal injury and edema that results in further neuronal loss, which worsen neurologic deficits and can also hamper recovery. The underlying mechanisms associated with this secondary injury and associated parenchymal edema have been sparsely described and not well understood in whole. As a result, current treatments are limited to supportive measures, management of intracranial pressure, and recanalization. Our goal was to understand the distinct role of microglia, monocytes, and neutrophils to this process, and as such be able to intervene in a targeted fashion. We were able to describe temporally distinct immune responses that can contain, exacerbate, and repair cerebrovascular injury. Using that knowledge, we developed anti-adhesion immunotherapy strategies to prevent fatal cerebral edema in a model of hemorrhagic stroke. Finally, we demonstrated why indiscriminate immunosuppression is detrimental in the setting of cerebrovascular injury. Using intravital microscopy following cerebrovascular injury in a mouse cortex, you can see here the differences between an uninjured normal cortex with a normal layer of astrocytic enphyte and an injured mouse where not only the vasculature is destroyed, allowing for a rapid influx of myelomonocytic cells, but also the astrocytic enphytes have completely retracted, not supporting the blood-brain barrier. We developed a sonication model of cerebrovascular injury that allowed for consistent, reproducible injury of the mouse cortical vessels. Our first observation involved microglia seen here in a normal uninjured cortex. Following cerebrovascular injury, the microglia rapidly created these tube-like structures within 10 minutes following the injury. These structures, I'm going to show in the next slide, surround injured vessels. I'm going to skip several steps of experiments and describe the conclusion about what the mechanism underlying this phenomenon is. The astrocytic processes, once injured, send an ATP signal through a connexin hemichannel to the extracellular space. This is being picked up by the microglia through a P2Y12 receptor. That leads the microglia to extend their processes and eventually migrate to the area around the blood-brain barrier, thus plugging the holes that have been created by the injury. Using inhibitors and the power of transgenic models, we were able to stop this phenomenon, and you can see here on the bottom left that if you prevent the microglia from resetting, you have extravasation of Evans blue at a much higher rate as compared to when the microglia are able to buttress the blood-brain barrier effectively. Similarly, you can see that in the case of small petechial hemorrhage that occur from this injury, if you again prevent this phenomenon from happening, you have much more diffuse, not contained intracerebral hemorrhage. This leads us to the conclusion that this phenomenon leads to the microglia rapidly reconstituting the blood-brain barrier after injury, as well as allowing for containing of intracranial hemorrhage. Our second observation is something I alluded to previously, and that is the rapid influx of monocytes into the parenchyma after the initial insult. You can see this here in one of intravital microscopy videos. This led us to find a way to stop this influx, prevent edema, and stop fatal cerebral herniation, as I would show in the next slide. We used a potent anti-adhesion antibody cocktail consisting of anti-LFA1 and anti-VLA4 antibodies. This has been studied extensively in other disease entities such as multiple sclerosis and cerebral malaria. Using this cocktail in a single dose, we were able to completely stop extravasation of myelomonocytic cells into the area of injury, as you can see here. More importantly, mice treated with a single dose of our antibody cocktail within 6 hours of the initial injury demonstrated a dramatic decrease in the accumulation of cerebral edema compared to the untreated mice. This single dose was enough to prevent cerebral herniation and allow for 100% survival after only a single dose of these molecules. Following this injury into chronicity, we were able to identify a delicate interaction between pro-inflammatory monocytes and angiogenic VEGF-positive microglia that was essential to orchestrate repair. Importantly, prolonged inhibition of the innate immunity disturbed this interaction and prevented angiogenesis and led to fibrosis. You can see here that in control mice, there is robust angiogenesis in the area of injury, and a majority of the neuronal population is preserved after the initial injury with only a small area of scar. In contrast, chronically immunosuppressed mice demonstrate inability to achieve any type of angiogenesis, and the area of injury and the neuronal population are replaced by a gliotic and collagen-rich scar. Bulk sequencing demonstrates separate clustering of chronically immunosuppressed mice compared to uninjured or controlled injured mice. These mice demonstrate upregulation of several key fibrotic genes. Importantly, the lack of angiogenesis associated with prolonged immunosuppression promotes neurological dysfunction and hampers recovery. Control mice 10 days after an initial injury demonstrate some motor and cognitive dysfunction, but slowly recover over the next 10 days, approximating control at 20 days, whereas animals that have been continuously immunosuppressed since the day of injury are not able to recover at all. To summarize, we described several temporally distinct immune responses that can contain, exacerbate, and repair cerebrovascular injury. This allowed us to develop an anti-adhesion immunotherapy strategy that can prevent fatal cerebral edema and shows promise for further therapeutic trials. Finally, we show how indiscriminate prolonged immunosuppression is detrimental and can hamper recovery following cerebrovascular injury. I would like to thank Dorian McGavern, who was my PI and mentor at the NIH, as well as all of my other collaborators without whom this work would not have been possible. For more details, you can find our recently published papers online. Thank you very much.